Hey, everybody wants to do this, Mr. Yance again. I'm going to just pick up where we left off last time. And I kind of like to start with this a little time lapse. We finished up with mitosis the other day, so I just kind of want to show you a quick video that shows a little time lapse and I'll kind of talk in the background as we go through this. So watch the phases as the cell goes through metaphase or prophase, metaphase, anaphase. Okay, so you should start to see some of the things we talked about last time in notes. Okay, that's the idea anyway. So this is sped up quite a bit, which has to be because this takes a little time, right?
The cell cycle of eukaryotic cells, which is typically much longer than that of prokaryotic cells, is divided into two major phases, interphase and mitosis, or the M phase. Interphase is the stage in the cell cycle in which a cell acquires energy and nutrients from the environment, grows, and duplicates its chromosomes. During mitosis, the chromosomes duplicated during interphase are separated to form two new nuclei. The later stages of mitosis are usually accompanied by a division of the cytoplasm into two daughter cells in a process called cytokinesis. Let's look at each of these phases in turn, starting with interphase. Cells often spend 90% of, or even their entire lives, in the interphase portion of the cell cycle. Throughout interphase, cells acquire energy and nutrients from the environment, grow, and carry out their specialized functions. However, a number of significant events occur during interphase that has caused biologists to divide interphase into three subphases. G1, which stands for first gap, S, which stands for synthesis stage, and G2, which stands for second gap. G1 is the phase after the most recent cell division and before the duplication of chromosomes starts. A large percentage of the cell's growth usually occurs during the G1 phase. However, some cells, such as the first cells to divide in animal embryos, do so very rapidly, with little or no growth between divisions and virtually no G1 phase. Later in the G1 phase, at a time referred to as the restriction point, cells undergo an internal evaluation of their ability to complete the cell cycle and produce two viable daughter cells. If the evaluation is negative, the cell does not go on to the mitosis phase, but rather exits the cell cycle to enter a non-dividing state called the GO phase. If the evaluation is positive, the cell becomes committed to DNA replication and mitosis. The restriction point can also be thought of as a point of no return for cell division. If a cell is going on to mitosis, it begins to produce proteins called histones that are critical to organizing and condensing chromosomes during mitosis. As the production of histones peaks, the S phase of interphase begins. During the S phase, duplicate copies of each chromosome are synthesized. This process can take quite a while, as many eukaryotes have a large number of very long chromosomes. For example, human cells have to duplicate 46 chromosomes, each 1,000 to 5,000 times the diameter of the nucleus in length when stretched out. After chromosomes are duplicated in the S phase, G2, which is usually the shortest phase of interphase, begins. During G2, cells further ready for mitosis by...
Okay, so hopefully you saw a little bit about the G0, G1, S, G2, those things we talked about the other day, and then the cell progresses into all those dividing phases, okay? All right, we're going to change gears a little bit. So we're going to talk about the other process of cell division called meiosis. Okay, so here's the reason we have two different types of cell division. Good test question that we'll have on the test. Cell specialization. So mitosis is involved in, let's make blood cells. They'll do their things. Skin cells will do their things. Muscle will do their things. And the only way we can do that is to divide the cell and make more of them and mass produce them. That's mitosis. And understand, I think this is a critical part of genetics, which we're starting the unit. That's basically we're starting our genetics unit right now. Is that understand that the whole book of life is in every cell. So it's not like blood has a different DNA than skin. Skin's got a different DNA than bone. They have the whole instruction sheet. It's sort of like when I work on my tractor home, I have the whole manual that's about yay thick. But if I got a problem with my PTO or I got a problem with the brakes, I go to that chapter and look at that part of the manual. And just like in English, the English teacher is not going to say, here's a 35 chapter book, read all 35 tomorrow. You're going to read chapters 1 through 3, 4 through 7, 5, things like that. So that's pretty much what a cell does. Is it just reads the instructions that pertains to whatever it's supposed to be doing in the body's blood, skin, muscle, whatever. All right. Now, the other problem we have, though, is we have these special cells, sperm and males, and ova, egg, and a female. And with those gametes, we call them, we can't have mitosis. I can't have the chromosome number stay the same. So let's look at reproduction because it's going to change the game a little bit. So let's start with a very simple one called asexual reproduction. Asexual reproduction is where I have one parent. I'm going to just basically carbon copy the DNA, and I'm going to make mass quantities of myself. So bacteria, mold, algae, protozoa, very simplified organisms, simple ones will use this type of reproduction. There's 4 of these. And again, another good test question. There's fission, which is the simplest form where I have one cell and then I have 2 equal cells. Okay, that's called binary fission. We're basically just going to split in 2. The downside to asexual reproduction is that if I have a problem, I have bad genetics, well, I'm going to pass that on to every single cell from that point forward because I'm just carbon copying the information. So you don't want to have bad genetics in a single cell organism, right? That's what's good about our genetics is we can block that, we can keep that from happening because we have 2 cells coming together. The plus side is they can reproduce really, really fast. Strep, pneumonia, you use those 2 for an example. They can duplicate and reproduce every 20 minutes really, really fast. Okay, so fission's the simplest form of that. You can see that up here. I got a copy of my DNA. They still go through. It's pretty much mitosis. They go through G1S, G2, and then they divide. But the difference is I'm making whole organisms, not body cells here. More bacteria. All right. Another type that we see is spores, where we duplicate the DNA and we get this little spore that gets released and it falls and grows into a adult. That would be another form of asexual reproduction. Budding is where we just pinch. Here's a hydra over here, and let me get my face out of the way. So you can see here's the adult. Here's a little mini me, a little small version, and it pinches off and grows. These are spores that you see, especially in mold and things like that. All right. Understand something. I do want to bring this point up because I think it's a valid point, and that is, bread mold. Let's take that for example, right? The problem with bread mold is you get a little spot, and my mother was very guilty of this in the past, and there'd be a little piece of mold on a piece of bread or a piece of cheese or something like that, and she literally would just cut it off and eat the rest of the bread, eat the rest of the cheese. It's just on that little piece that I can see. Not true. With spores, you don't know where those have landed. So even though it may not be the adult green, blue mold you see growing on the other part of the bread, it's still there. Just a good idea just to get rid of it. Okay, not a good idea. Just cut it off and eat it. All right. Those are the 3 natural ones: budding, spores, and fission, or the common asexual reproduction you see in a lot of the lower forms of animals and plants. Cloning is a man-made one. So with cloning, we're taking a cell, not a sperm or egg cell, just a body cell. We take it out of the body, put it in a nutrient compound, let it grow, let it divide, and put it in a surrogate mother, and we carbon copy that individual. Exact copy. Okay, so let me kind of show you that. I don't want to spend a ton of time on that right now because I have, we'll talk more about it later when we get into genetics, but let me kind of show you the process. So if you look at this news article I was teaching back then in 1997, I still remember looking at this and kind of scoffing and saying, oh, we're never going to clone a mammal, or we're way too complex. This not going to happen. And then we cloned Dolly. Dolly the sheep was the first mammal we ever cloned. And we're like, oops, we just ate our words there, and now it is possible. So this is Dolly. You can see this is the one that gave birth to her, but that is not really the biological mom. This is a clone. She just carried it and gave birth to it. It wasn't her baby. Okay. Now, understand cloning. So why did we do this other than to say, hey, we cloned a mammal? That's awesome. We have a lot of restrictions in this country. We don't allow cloning research for humans. And the reason for that is because we don't see a benefit from it. There seems to be a higher risk of diseases and shorter lifespans and things like that. So we said we're not going to clone a human, but other countries are trying to. So, and I'm sure it'll happen. We've already cloned a chimpanzee, so we've cloned some pretty higher forms of organisms. And the idea about Dolly here is in agriculture. Let's say here's the idea. We have this champion sheep. It has great wool, wins state fairs, is really valuable, worth a lot of money. Why don't we just clone it? That way I get the same one over and over and over again, and I keep getting a grand champion sheep. Paper. Problem is, it took hundreds of thousands of dollars and 10,000, let me say that again, 10,000 tries to get Dolly. Not very practical and not going to be a good idea. All right. So how that works, let me kind of show you the process really quick here. So the black-faced sheep you saw there was called a Scottish black face. She's down here. And then this is called a Finn Dorset, which has a white face and white wool. So what you do is you take a mammary cell, isolate it. You can see it here. And we're taking it out of this sheep, not an egg or sperm. Okay. We take an egg out of the surrogate mother here, the black-faced Scottish blackface, and we pulled her DNA out. So you have this oocyte, which is really an egg with no DNA. It's empty. Okay. Then we come over here, and you see a lightning bolt here. We call this Frankenstein technology. If you guys know about Frankenstein, the story, he comes to life from a bolt of lightning, right? This DNA will not go into this oocyte unless we make it. So we have to zap it with electricity and force it to go in there. So here's what we have. We have an egg out of this sheep with the DNA from this sheep. Okay, that's cloning. Put it in a petri dish, let it grow, let it divide, put it back into the surrogate mother. She gives birth not to her baby, but she gives birth to a clone of this one up here. That's pretty much how cloning works. It's kind of fascinating. This is showing you how they zap the DNA into the empty egg. Okay, so cloning's been around a little while now. You see what we've cloned so far: dogs, cats, sheep, deer, horses, chimpanzees. Just recently in China, and this is kind of a cool video. But I'll explain it a little bit when we get done. So here we go. So listen to this.
Have you ever thought about cloning your dog or cat? Now you can at myfriendagain.com. The process is simple. Call us and we'll ship a biopsy kit to your vet. Your vet will extract a small sample from your pet and ship it back to us. Our scientists will use that sample to create a clone of your pet. Learn more at myfriendagain.com.
Okay, sounds great. I got my beloved dog. I have one. Sasha's her name. She's getting sick. She's getting up there in age. Let's say she's getting old. I want to get a copy of her so I can have her for some more years. So I send my sample to this place. What they don't tell you is when you get the more information it costs $10,000 just to ship the sample. Let me say that again, $10,000. And then you have all the costs of cloning. So, I love my dog. I love my pets. But I'm not going to spend $20,000, $30,000 to get a clone. Okay. And understand something. A clone is a carbon copy of your beloved pet, my friend again, but it's not the same dog or cat. Dogs and cats have personality. That's not going to be the same thing that you may get in your clone. So you may not get exactly the same dog and cat you think you're going to get. So kind of remember that. Okay. So, that's kind of where we're at in cloning. Fascinating stuff. A lot of controversy with it, but it is possible to make carbon copies of the same organism. All right. So that's asexual reproduction. Let's talk about sexual reproduction, where I have two individual two different cells coming together, an egg and a sperm. They're called gametes, and I get a zygote, which means to join. Now, the problem is going to be if I use mitosis, I'm going to get 46 and 46. I'm going to get, 92 chromosomes. That's not going to work. So you have to get that chromosome number reduced. And that's why we have this special cell division called meiosis. It's only in an egg or sperm cell. That's the only place it occurs. It's only involved in sexual reproduction. So that's what we're going to talk about in the next set of notes. So hopefully that makes sense. So have a great day. If you got questions, again, email me, let me know. So we'll see you. Bye.